Okay, genetics, fun facts, and trivia. Here we go. First, haploid and diploid numbers for different organisms. You can see on the table that there's a big range of chromosome numbers. Different organisms have different numbers for their haploid-diploid number. Kids might think that there's some relationship between how big an organism is and how many chromosomes it has, and there really isn't much of a pattern to it. The tiny little butterfly has 268 chromosomes in its cells, and the big kangaroo has 16. We mostly look at plants and animals for these numbers because they're the ones that sexually reproduce, so haploid and diploid mean something. There are some organisms that regularly exist as tetraploid. There's some kind of mouse that walks around with 4n as its number, but I just kept it simple here with haploid and diploid numbers for a bunch of different common organisms, and you can see the different ranges, but no association between how many they have with how complicated or large the animal or plant is. Crossing over and meiosis. Here we have first the timing of meiosis. It takes a lot longer than mitosis, and the big reason is because prophase 1 in meiosis takes a long time. Also, prophase 1 is where crossing over happens. In humans, meiosis in males can take 20 to 25 days. Females, it kind of starts and then gets delayed, so there's a developmental hold on meiosis, and one cell division can take up to 40 years technically. But even fruit flies, where mitosis is fast, meiosis is 1 to 2 days long. We have theories that explain crossing over in terms of DNA repair mechanisms. That's where it's thought to have originated. And on the right there, you can kind of see a little diagram I put together that shows 3 different traits, or the genes that control 3 different traits that are all on the same chromosome. B and C are close together on a chromosome, and A is further away. Well, the way that we figure out how far apart things are on chromosomes is by studying how common those traits are in offspring. And based on the phenotypes of the offspring, you can use some fancy math to estimate how many base pairs separate 2 traits. So for example, traits B and C are probably going to stick together. So there's not likely to be a crossing over event that happens in between B and C because there's only a small region where they could cross over and get separated. A and C or A and B are much more likely to get separated by a crossing over event, so we know that they're further apart on the chromosome. All that sounds kind of fancy and complicated, but if kids take a sophomore level college biology class, like a genetics class, they'll probably do a fruit fly genetics lab where just based on counting fruit flies with different color eyes or different shapes in their bodies, they can actually make these linkage maps where you count how many base pairs, or you calculate how many base pairs separate different genes. I remember doing it in sophomore genetics class, and we all went through the process because we were supposed to, and nobody expected that we'd actually get numbers that were correct. But sure enough, a couple of different traits that we just counted offspring for gave us numbers that were very close to the actual lengths of base pairs separating different traits on some of the fruit fly chromosomes. All right, we'll learn more about this when we go to the Sam Ryan conference on February 2nd. Remember to put that on your calendars, but most traits that we study in biology are kind of uncommon things that we pretend are common so that we can learn the principles of genetics. We pretend that things are controlled by a single gene, and that gene has 2 alleles, and one of the alleles is completely dominant and one of them is recessive, and that helps us get kids to understand some of the basic principles of genetics. But most traits are polygenic, so controlled by many, many genes, and a lot of those genes have multiple alleles, so not just two alleles. Genetic research now is getting better at figuring out things that are controlled by more than one gene, and even common traits like eye color, hair color, height are being studied. And from sources I found, there are about 15 or 16 different genes that are thought to influence eye color. There's a couple hundred. One source said about 240 or 250 genes that affect height. Skin color, I think, is determined by about 4 or 5, and they're basically all co-dominant, so different levels of pigment give us all the different skin colors. And you can see that really complicated looking Punnett square over there has all the different levels of melanin producing cells in people, in offspring, to give us all different shades of humans. Heteroplasmy is kind of an interesting thing. What you're looking at there is a representation of mitochondria in a maternal cell, and then there's all different sorts of possible ways to split up those mitochondria to make offspring. So inside mitochondria, we know there's DNA, so everybody has, or every cell has, a mitochondrial genome, the same way you have a nuclear genome. So the mitochondrial DNA can mutate just like any other DNA, and if there's a mutation in mitochondrial DNA, that mutation will only be inherited by some cells, because if there's a mutation in one mitochondria, it's only going to go to one of the two offspring in meiosis. Once that cell starts to replicate, that mutation starts to replicate, and the mutation will continue to spread throughout the different generations of offspring of the cells. So what you end up with is a whole bunch of different cells that all have different percentages of healthy versus mutated mitochondria. And heteroplasmy refers to the fact that you have lots of different compositions of cytoplasm, so different cytoplasms in all your different cells. And if that mitochondrial mutation is something harmful or unhealthy, the health of a cell might depend on what percentage of mitochondria are healthy versus what percentage are mutated. A little bit of biotechnology here. PCR, polymerase chain reaction, was discovered in the 80s. 1983, by that guy whose picture you see over there. His name's Kary Mullis. He's a kind of a weirdo. He's got some crazy stuff in his career, one of which is that he used a lot of LSD, and he claimed that he had visions for how PCR would work due to his use of LSD. But he won a Nobel Prize for it, and we wouldn't be able to do a lot of the research that we do these days without PCR. Basically, it's a way to amplify a section of DNA to make many, many copies exponentially. And it's really simple once you have the starting material. So you need the piece of DNA of interest, and you need RNA primers, so short pieces of RNA. And then you need an enzyme to make more DNA, and that enzyme's DNA polymerase. But it's not just any DNA polymerase. It's a certain kind called Taq polymerase that comes from the bacteria that you see here. The DNA polymerase is extracted from these bacteria that are thermophilic, and they live in hot springs. Well, their DNA polymerase survives really, really high temperatures. So to do PCR, you put the DNA of interest, the RNA primers, the DNA polymerase, and a whole bunch of nucleotides together in some sort of reaction chamber, and you heat it up and you cool it down, and you heat it up and you cool it down, and you heat it up and you cool it down. And all those cycles of temperature change cause the different generations, I guess, or the different amplification stages of multiplying that piece of DNA. So PCR is a pretty cool technique, and we wouldn't be able to do much of biotechnology today without it. Another thing really important to biotechnology are restriction enzymes, and those are the enzymes that bind to specific sequences of DNA and cut it into pieces. They all have a recognition site where they bind to that specific sequence. Sometimes they cut the DNA at that recognition site. Sometimes they bind to one site and cut the DNA at another site about 1,000 base pairs away. They come from bacteria too. Bacteria use them to protect themselves from viruses. So viruses that come in and use DNA as their genetic material, the restriction enzymes can chop up that DNA. And they don't chop up the bacteria's DNA because the bacteria has another enzyme that protects its DNA from its own restriction enzymes. A lot of the restriction sites for some reason are palindromic, so they have kind of this symmetry to their sequences. I don't know why. Restriction enzymes were discovered in the 50s and then into the early 60s. The first kind they discovered were the ones that chop up the DNA at the site 1,000 base pairs away from the recognition site. But then in the 60s, they found ones that cut the DNA at the recognition site. Those are the ones that are really, really useful for research, because all you need to do is know a sequence of a bacterial plasmid or of any piece of DNA, and you can predict exactly what pieces it's going to get chopped into if you mix it up with some restriction enzymes. The names of restriction enzymes get assigned based on the bacteria that they get extracted from. And again, it's one of those things that we were lucky to figure out because it enabled all the kinds of biotechnology research and manipulation of genes that we do today. You guys have probably seen these all before. It's just a quick little summary, very brief summary of chromosomal disorders in humans. So having extras or missing a few chromosomes. I think we've heard of all these before, but there are some really rare ones with the sex chromosomes, like 4 X's and a Y, 2 X's and 2 Y's. They're really, really rare. Some of them have only ever been observed in 30 or 40 individuals ever. All of those also have really varied phenotypes or symptoms. But this is just a good little summary that can kind of put together all the different chromosomal disorders with a really brief description of what they do to offspring. A lot of these are kind of sad, so kids could ask questions and want to see pictures, but they're pretty rare, and they're not super important, other than obviously Down syndrome, which is the one that kids are most likely to come across. But they'll ask all sorts of questions about the ones with the sex chromosomes, and they'll come up with some tough questions that are probably hard to answer. So I wouldn't spend too much time getting into all the nuts and bolts of these. Just make sure they understand how they happen and that they're all generally pretty rare.